Now, as I talked about in the last lecture, Lick protein that is associated with CD4 or CD8. Once CD4, CD8 becomes interacting with the MHC, stable, changes its cytoplasmic tail, which will ultimately result in the activation of Lick. Lick will then go and start the phosphorylation process of ITAMs in its immediate vicinity. The ITAMs recruit in ZAP70. ZAP70, when it becomes active, then goes and activates some different scaffold proteins, LAT and SLP76. LAT and SLP76 recruit in and activate phospholipase C gamma. And what this looks like. CD4, CD8, depending on which T cell you're talking about, it has this zinc finger here and its cytoplasmic tail that has a SH3, SH2 domains at its terminal end. But notice there the SH2 is phosphorylated. If it's phosphorylated, it is holding Lick in an inactive state. Becomes dephosphorylated when signals come through other receptors. Here specifically they're showing the example of CD45. CD45 then releases that phosphate group, which then allows Lick to become an inactive form. And it goes and starts phosphorylating ITAMs in its immediate vicinity. The ITAMs become phosphorylated, so you get the CD3, sigma, delta, gamma, and epsilon. So you end up with these becoming phosphorylated, which will recruit ZAP70. ZAP70 bound to a phosphorylated ITAM will then phosphorylate, activate SLP76 and LAT. SLP76 LAT brings in the protein GADs. GADs and all these complex recruits in phospholipase C, which will then be activated by ITK, which is bound. And phospholipase C, if you've had me for other courses, you know I say that then becomes the lawnmower and just starts cutting all the PIP2s, releasing IP3. So the activated PLC, it goes through and releases second messengers. Diacylglycerol is not a second messenger so much because that is basically the glycerol and the two fatty acids that remains in the lipid bilayer. But what gets released is IP3. IP3 will then go and will bind to gated ion channels in the ER, releasing calcium. Calcium channels get opened and then they flood through. Flood out of the ER to the cytoplasm or from extracellular in. You take a closer look at this. You see phospholipase C goes, cuts. IP3 will bind, crack, or gated ion channel. The crack will allow extracellular calcium. The gated ion channel here allows those in. So what happens is the gated ion channel opens because IP3 binds to it. It's the key to opening it up. Calcium floods through. You end up with certain proteins being, binding in, which will then trigger other proteins, PKC, RAS, GP, to be bind to diacylglycerol, which is still stuck in the lipid bilayer, which will call these crack channels. I just love that name, crack channels. Crack channels to open, allowing the calcium from the cytoplasm either to flood out or, if there's high levels of extracellular, to flood in. And all because, as we saw in the last channel, or the last couple of slides, CD4 became stable while it was bound to the T cell receptor and MHC were interacting, which allowed for the activation of LIC, phosphorylates ITAMs. Here they're showing T cell receptor CD3, the complexes of CD3 around it, ZAP70, LIC, and there's an accessory one here known as FIN, which I didn't talk about. And you can see it starts off with one signal that can branch off. IP3, the diacylglycerol. Diacylglycerol can activate multiple things just because of its mere presence there. It acts as a landing pad, recruits in other signal proteins, which, because they're now bound and stable where they're at, can interact with other proteins, and it starts alternate domino pathways falling, other signal pathways falling. Ultimately, all of it ends as transcription factors, upregulation of certain expression of certain genes, downregulation of others.